Very good morning to all of you. So, students, today we will study about the chapter that is cell chapter. This is basically for class ninth. Okay, this is for class ninth, and according to the new book that is Exploration, the name of the chapter is Cell: The Building Blocks of Life. So, let's start with this particular chapter. So, children, you can see on this particular screen that the cell is basically discovered by a famous scientist named Robert Hooke. The name of the scientist was Robert Hooke. So Robert Hooke has discovered the cell which is the living cell, not the dead cell. Okay. So Robert Hooke has discovered the living cell. Now you can see here on the next slide that there is a theory called as the cell theory. Okay, cell theory is given by two scientists basically, Matthias Sheldon and Theodore Schwann. And one of the scientists named Rudolf Virchow, who had added one statement here. The question used to be asked, what is that statement added by the Rudolf Virchow? So the answer is all cells arise from the pre-existing cell. This is the statement added by the Rudolf Virchow. You can see on this particular screen, there is a cytoplasm, nucleoplasm, and protoplasm shown. So suppose this is the cell. Whatever the liquid, the jelly-like liquid present inside the nucleus, this is the nucleus, is called as nucleoplasm. Remember this thing, the jelly-like substance present inside the nucleus is called as nucleoplasm. So here inside this nucleus is nucleoplasm. It includes the nuclear material also, that is the genetic material also, and the jelly-like liquid present between the cell membrane. This is the cell membrane, suppose. Okay, this circumference of this particular cell, suppose it is circular, and between this cell membrane and this nuclear membrane, so whatever this blue color liquid is there, sky blue color liquid is there, that is a jelly like substance called as the cytoplasm. And both this cytoplasm and nucleoplasm collectively called as protoplasm. So we used to say that whatever the jelly like substance present inside the cell is protoplasm, or we used to say that the living component of a cell is called as protoplasm. So this is the diagram of prokaryotic cell. Prokaryotic cell has the example called as the bacterial cell. Okay, you can see that the bacterial cell is composed of three different layers. One is the capsule, that is the protein capsule. That is the cyst, which is a protective membrane and extra layer of protection given to the bacterial cell called the capsulated layer, which is formed of protein. Then we have a cell wall, and the third one is the cell, that is the plasma membrane or the cell membrane. So there are three layers of protection present inside the surrounding the prokaryotic cell, that is the bacterial cell. Then you can see here the naked DNA is present. Like in the previous slide, I told you about the nuclear membrane. Okay, so this was the nuclear membrane. This nuclear membrane is not present in this cell called as a prokaryotic cell, which has the example bacterial cell. Rather, DNA is naked. Naked DNA means without nuclear membrane, and they have the free protoplasm, free. We used to say ribosomes are there. So these are the crystalline circular structures present in the bacterial cell is called as the ribosomes. Ribosome have the function to secrete the protein or the production of protein. Okay, one more important thing here you can see on the screen, the region called as the nucleoid region is shown. I told you that the nuclear membrane is absent in the prokaryotic cell. So the nuclear membrane is absent, whatever the region which contains the genetic material is called as the nucleoid region. So we used to say that this DNA, the region where this DNA is present, the genetic material floats freely in a single circular molecule without a protective nuclear membrane is called as the nucleoid region. Okay. So this is all about the bacterial cell. Now you can see a comparative account of animal cell and plant cell here. And this plant cell and animal cell are the examples of the eukaryotic cell. Okay, so you can see a comparative account here. The eukaryotic cell, that is the animal cell, contains basically a central nucleus. Rather, in plant cell, the nucleus is peripheral in nature. So what is the meaning of peripheral? Peripheral means corner nucleus is there. Why do the nucleus shift towards the corner in the case of plant cell? So because of the presence of large vacuoles. So this is the structure, that is a large vacuole is present in the plant cell. That is why the nucleus is shift towards the corner. Now the question arises, why do the plant cell contains a large vacuole? So vacuole basically has the function to store the food. Okay, so plant cell needs to store the food. Animal cells do not need to store the food. Why? Because animals are mobile. They have the mobility, they have the feature of locomotion. They can go here and there in search of the food, but plant cannot go here and there in search of the food. They have the limited time for the production of food. That is why they need to store the food for the time when the food cannot be available, or we can say that when the sunlight is not there. Okay, so plants used to do the photosynthesis at the daytime, and they used to store the food in the vacuoles. Now you can see here, this is the chloroplast, this green color structure. Okay, this is the double membrane green color structure is called as the chloroplast. Although whole cell is looking green, but here the exact greenery you can see here in this cell organelle called as the chloroplast. So chloroplast is called as the kitchen of the cell, and again it is only present in the plant cell only, not in the animal cell, responsible for the photosynthesis, that is the production of food. Okay, now you can see here there is an extra layer of protection in the case of plant cell, that is the cell wall. In the animal cell, there is a single layer of protection. You can see there is only one layer of protection, that is called as the cell membrane. But in the case of plant cell, cell wall and cell membrane both are present. So this is the cell membrane, that is the second layer, and cell wall is the first layer. So both of these are present. Now you can see here in this animal cell what are the unique features present in the animal cell only. As the first point we had already discussed, that the central nucleus is present because the vacuoles are absent in the case of animal cell. Again, lysosomes are present. Lysosomes are the suicidal bags of the cell. They have the function to secrete some digestive enzymes so that any bacterial infection which comes into the cell will get killed up. Again, if the bacteria infects the whole cell, in that case the lysosome bursts and kill its own cell. The process is called as autolysis. Auto means self, lysis means breakdown. So lysosome can also be called as the atom bomb of the cell. They get burst out in the case of bacterial infection and kills the whole cell. Sometimes they destroy the dead and worn out cell organelles also. Means if infection is not very much larger, so in that case the lysosome will burst out and release some of the digestive enzymes to kill its cell, the worn out cell organelles only in that case. Now come to the next point, that is you can see here an comparative account I had written here about the plant and animal cell, that we had already discussed about the presence of cell wall, plastid, basically the chloroplast, and vacuoles. Now you can see here in the next slide, that is of the structure of nucleus. You can see, so nucleus is also called as the brain of the cell, control center of the cell, director of the cell. The nucleus contains a double layer of protection, that is the nuclear membrane. And you can see this is the nuclear membrane, and after a certain amount of or the distance, there is a nuclear pore present. So you can see these are the nuclear pores. What is the function of nuclear pores? Nuclear pores allow the substance to pass through the nucleus, and whatever the nucleus needs to communicate with the cytoplasm, it takes the help of this nuclear pore, and the substance go from nucleus to outside, that is in the cytoplasm, or if anything wants to come from cytoplasm to nucleus, it can comes in the nucleus also. There is a non-nuclear structure present, or we can say a non-membranous structure present. Non-membranous means without having any membrane. This is called as nucleolus. You can see here, we had mentioned here, a dense body where ribosome subunits are manufactured. Basically, inside the nucleus, there is a non-membranous structure is present. Non-membranous means without having any covering. That is called as nucleolus, which contains the ribonucleic acid, that is the RNA. And what is this rest of the structure which is present, which is a supercoiled structure? There is a spirally arranged a supercoiled structure is present, that is called as the, basically this is the DNA, but we used to write it as the chromatin because the DNA is a double helical structure. It has a proper double helical structure. So this is the chromatin material present, and this chromatin material basically composed of two things. One is the DNA, another one is the protein. So protein is also embedded here. Okay, so that is all about the nucleus. Now you can see here in the next slide, mitochondria. Mitochondria basically the powerhouse of the cell, and it produces power or the energy that is in the form of ATP. This is the adenosine triphosphate, that is the final energy of the body. If somebody asks your grandmother how you will get the energy, the grandmother used to say we get the energy by the help of food. If somebody asks any other person who had read about the glucose is the instant energy. So he or she used to say that the food get converted into glucose, and the glucose gives us energy. But now from now onwards, if somebody asks you people that how you will get the energy, you have to say the food gets converted into glucose, and glucose in turns converted into ATP by the help of mitochondria, and that is the final energy currency of the cell. If somebody is converting food into glucose and not into ATP, means food can be converted, can easily be converted into glucose, but the glucose is not able to get converted into ATP. The person will feel lazy or non-energetic, and it actually happens in case of diabetes. In the case of diabetes, what happens, your food get converted into glucose, but the glucose could not be able to convert it into ATP. In that case, the person used to take the injections of insulin, which help the glucose to get converted into the final energy compound. Okay, so that is actually the thing. Is one more very important thing: the mitochondria basically contains its own DNA and ribosomes. DNA and ribosomes. So DNA and ribosomes presence help this particular structure mitochondria to function by its own. That is why it is also called as semi-autonomous cell organelles. Autonomous means have the autonomy, or you used to say that they can control themselves. So nucleus can be the complete autonomous cell organelle, but the mitochondria is semi-autonomous because it can control some of its processes because of the presence of DNA and ribosomes. Remember this thing. And not only this particular mitochondria, but we'll study after this particular slide about the chloroplast also. That is also a semi-autonomous cell organelle because the chloroplast also contains its own DNA and ribosomes. Okay, so it's all about this mitochondria. One more thing is there, that is this particular inner membrane. You can see this is the outer membrane, and this yellow color inner membrane is present. What is all about this particular inner membrane? Inner membrane is responsible for the production of energy, that is the ATP. Okay, and you can see there are folds present on this particular inner membrane. These folds are called as cristae. Why do the inner membrane is folded in case of mitochondria? So remember this thing: mitochondria has the inner membrane which is folded so that the length of the inner membrane should be increased, because the function of the inner membrane is the production of energy. Okay, so we want more energy to be produced. That is why the inner membrane is folded. Okay, now you can see, come to the next slide, that is of the chloroplast, that is the kitchen of the cell. Okay, again, this is the double membranous structure responsible for the process of photosynthesis to take place. Okay, and you can see these are the disc-like structure present in the chloroplast. This disc-like structure are called as thylakoids. Thylakoids contains the green color pigment called as a chlorophyll, responsible for the absorption of sunlight for the process of photosynthesis to take place. This matrix, or the, we can say that the empty space, is called as stroma. Okay, so this is the stroma is there, and thylakoid and stroma, this disc-like structure in this empty space, or we used to say that stroma is basically both are responsible for the production of food, that is the glucose. Okay, again there is the self-sufficient cell organelle, that is chloroplast, because it contains its own DNA and ribosomes. So this is basically all about the chloroplast. And there are certain thing, one more thing is I can see here on the screen, that is the granum. What is this granum is all about? These are the stacks of thylakoids, okay, or the bunch of thylakoids. So this is called as the granum. And this is the lamellae or lamella, which connects a bridge like structure is there between the thylakoids, is called as lamellae. Okay, so that's all about the chloroplast. Coming to the next structure, that is the endoplasmic reticulum. Endoplasmic reticulum, the name suggests endo means inside. Okay, and basically it is endoplasmic reticulum. So okay, so this is present inside the plasma membrane, and it is a network like structure. Okay, reticulum means network, endo means inside, and plasmic means plasma membrane. So this is the network like structure present inside the plasma membrane, is of two kinds. One is smooth ER, and another one is rough ER, rough endoplasmic reticulum and smooth endoplasmic reticulum. Why do it is rough? Because on the surface there are certain structural components, you can see crystalline components, you can see these are the ribosomes responsible for the protein synthesis, and smooth ER do not contain this kind of ribosomes. That is why its surface is smooth and its surface is rough. So, what is the function? Smooth ER has the function of synthesis of lipids, that is fats, and the rough ER have the function of synthesizing protein, because definitely on its surface ribosomes are present. So ribosomes have the function of synthesizing proteins. So we used to say that rough ER also have the function to synthesize proteins. Okay, now you can see there is a structure called as the Golgi bodies. Golgi bodies have the function of basically packaging protein and lipids. As in the earlier slide, previous slide, we had studied that the smooth ER and rough ER forms the lipid and protein respectively. So this lipid and protein, which comes from the endoplasmic reticulum, will go for modification and sorting out, and finally for packaging in this Golgi body. So Golgi body is again contains some tubules which are called as cisternae, and these are the cisternae, or the tubular structures are there, and these are the vesicles. So whatever the packaging occurs, it occurs inside the vesicles, and these are the cisternae or the tubular structure. So the lipid and protein which is formed by the endoplasmic reticulum will enters from the cis phase. Cis is for cisternae phase of the Golgi bodies, and after packaging they comes out from the trans phase. Trans means transfer phase of this Golgi bodies, and they get transferred to their proper location. So you can see here these are some of the secretion outside the cell which can go outside the cell, become lysosomes. There are certain vesicles which becomes lysosomes also. Lysosomes, as we had discussed in the slide where the diagram of animal and plant cell was there. Animal cell contains the lysosomes. We had already discussed lysosomes are the atom bomb of the cell. They have the function to destroy the whole cell, or that is the autolysis, or sometimes they destroy the dead and worn out cell. Sometimes they removes or kills the bacterial cell also. Okay, so these lysosomes are also formed by the Golgi bodies. So the Golgi bodies or the packets which contains the some digestive enzymes, they becomes the lysosomes. Okay, digestive enzymes which kills the bacterial cell or the whole cell if required. Okay, and remember why do how they actually are making the lysosomes. So lysosome contains the digestive enzymes, and all enzymes are proteins. Okay, and protein is formed by the endoplasmic reticulum. So the proteins which have the digestive enzymes or which act as the digestive enzyme will become the lysosomes. Okay, in this way, this whole process is called as membrane biogenesis. What is called as membrane biogenesis? Okay, membrane biogenesis is the formation of membrane or the maintenance of the membrane, because the cell membrane is composed of lipid and protein. So whatever the lipid and protein is required for the formation of the cell membrane is formed by the endoplasmic reticulum itself, and endoplasmic reticulum that transfer that lipid and protein to the Golgi bodies for the packaging, and those packets in the form of vesicles will go to the cell membrane. In this way, the cell membrane has the structure composed of this particular by the help of this particular process. Okay, coming to the next point, that is the you can see here the ribosomes and lysosomes we had already discussed. Ribosomes are responsible for the protein synthesis, and lysosomes the cleanup system or the killing crew of the cell, or we used to say that the suicidal bags of the cell. Coming to the next, membrane biogenesis, we had already discussed that how do this lipid and protein is formed. That is the you can see this structure is of the cell membrane. It is composed of lipid and protein. Okay, this is the lipid and protein is there, and this is smooth ER and the rough ER will transport this lipid and protein to the Golgi bodies for modifying, sorting, and packaging. Those packets will go to the cell membrane, and in this way, this selectively permeable, or we used to say that the semi-permeable membrane is formed. Coming to the next point, that is the osmosis, diffusion, and active transport. This is also one of the very important topics of this particular chapter. What is diffusion? Diffusion means the from higher concentration to lower concentration molecules are moving, and that is a free movement. Okay, there is no membrane is present, but in the case of osmosis, water movement occurs with the help of the membrane. And again, from higher concentration of water to lower concentration of water, this actually moves. And in the case of diffusion, the solid, liquid, and gases, all three components can be moved. But in the case of osmosis, only the water actually moves. So this is all about the comparative account between the diffusion and the osmosis. Also, you can see one of the process called as active transport. In the case of active transport, the movement of molecules from lower concentration to higher concentration occur. So if you compare this active transport with the osmosis and diffusion, you will find in the case of diffusion and osmosis, the molecules are more in number on the left side and less on the right side. So the diffusion and osmosis both occurs from their higher concentration of molecules to lower concentration of molecules. So these are the lower concentration molecules. In the case of active transport, the molecules actually moves from the lower concentration to higher concentration. So if anything which goes from lower to higher, the energy is required. Again, that is in the form of ATP. ATP is the final energy currency of the cell formed by the mitochondria, that is the powerhouse of the cell. Okay, so this is all about the diffusion, osmosis, and active transport. There is an example of the active transport. When the plants needs to absorb the mineral ions from the soil, they used to apply the force to absorb the mineral ions, because the mineral ions basically is present in the soil particles comparatively, as the cellular structure in the lower concentration, inside the cellular structure it is in higher concentration. So as a whole, we used to say that the structural feature where the low from lower concentration to higher concentration movement occurs, that is called as the active transport. Okay, you might get confused with this particular point, that the soil always contains the mineral ions in higher concentration. Why the sir is saying that it is in lower concentration? So it is just a comparative account between that particular soil particle from where the cell needs to absorb the mineral ions. So if you compare the same structure of a soil particle with the same structure of the cell. Suppose the same volume we are asking about. Okay, we are talking about of the same volume. So as a whole in the soil, the mineral ions are more, but from where the soil particles is present, from the soil particle we need to absorb the mineral ion. So as compared to the same cellular structure and the same soil structure, the mineral ion is more in the cellular structure. Okay, so that is why we used to say that the ATP is required. Okay, so this is the tonicity. What is the tonicity? There are three kinds of solution. One is the isotonic solution, another one is hypotonic solution, third one is the hypertonic solution. Iso means equal, tonic means concentration of salt or sugar. Hypo means less, tonic means concentration of salt or sugar. Hyper means more, and tonic means again concentration of the salt or sugar. So in the case of hypertonic solution, the outside this particular cell, okay, in this particular solution where the cell is placed, the concentration of salt and sugar is more. In the case of hypotonic solution, the concentration of this particular salt is less, and in the case of hypertonic solution, the concentration of salt or sugar is more outside the cell. Okay, so the net movement in the case of hypertonic solution, water absorb inside. Hypertonic solution, the movement of water occurs from inside to outside. Okay, so this is all about the mitosis and meiosis, and this is actually called as the tumor. It's all about the tumor. Tumor is nothing but the uncontrolled division of the cell. Okay, so that is actually the tumor. That is the uncontrolled division of the cell. You can see this is the normal cells, and when any uncontrolled division occurs, that becomes the tumor. In the previous slide, that is the slide of mitosis and meiosis. The These are the two kinds of cell division. Just you have to remember that in the case of mitosis, which actually happens in the somatic cells or the normal cells, the meiosis happens in the case of reproductive cells. Okay, and so this is actually all about the mitosis and meiosis, and so this is actually the structure. One more thing is there, in the case of mitosis, two daughter cells are formed. In the case of meiosis, four daughter cells are formed. Number of chromosomes are 46 only in the case of mitosis, and in the case of meiosis get reduced to 23 or reduced to half. So that's all about then the normal explanation or the fast explanation of the cell. So thanks a lot. Thank you very much. I hope you like this particular lecture. So you can revise the lecture with the help of this particular video. So thank you. Thanks a lot. Thank you very much.